Okay. Hello, everyone. Welcome to our seminar series. If you don't know me, my name is Amadeo Salvador. I'm a postdoctoral fellow under Dr. Mentor of Dr. Elizabeth Parks. And today I have the honor to introduce Dr. David Bishop. I just want to make a side note that when Dr. Dillon asked me to introduce him, I immediately text my mentor from my undergrad mentor and my master mentor to say that I'm going to introduce David Bishop. I've been reading his papers for more than a decade right now, and he was really important for my undergrad thesis. So this is really nice for me. Professor David Bishop leads a team focused on optimizing the prescription of exercise medicine to improve health and performance. His team combines basic laboratory research with randomized controlled trials to address how exercise alters molecular pathway, skeletal muscle adaptations, and whole body metabolic health. And the goal is to translate this new knowledge into recommendations for more individualized exercise prescriptions and to better improve fitness and health. Professor Bishop has more than 300 peer-reviewed articles in the area of exercise and sports science, and his research is currently funded by the ARC, NHMRC, the MRFF, and the Australian Defence Force. He's also the past president of Exercise and Sports Science Australia. Please join me to welcome Professor David Bishop.
Thanks a lot, Amadeo. I'll try and share my screen. Okay. Okay, here we are. Sorry, I'll just need to... Oops. Sorry, everything's... There we go. Great. So hopefully you can all see that. I'll just get rid of that. Please, thanks for the introduction, and Amadeo, and thanks for the invitation to speak as well. It's great. We were talking before, so yeah, it'd be great to meet you all in person, but we've got the online option here. I don't know how these normally work, but feel free to interrupt me any stage if you've got any questions. Otherwise, I'm happy to save questions until the end. It was interesting to. Wasn't going to say this, but it was Amadeo reminded me. I guess a lot of my career started more as a sports scientist working with athletes, and a lot of my early research was on repeated sprintability, which won't get a mention here today. But I guess where I'm going to talk today and where my career has taken me is, I think, taking some of those principles from sport where the training is quite accurately and precisely prescribed, and just trying to see, if we can apply some of those similar principles to exercise so that we can get better adaptations from exercise. And so that's what I'll be talking about today, is how can we better prescribe exercise to improve health, but specifically to improve mitochondrial health. Okay. Now, I quite often start off with this slide, and I think most people are familiar with this or something similar. So this is from Plato, so nearly 2,500 years ago, and just talks about how lack of physical activity destroys the good condition of every human being, while movement and methodological exercise save it and preserve it. So, sometimes we think we're at the cutting edge, but, the idea that physical activity can help to improve health is a pretty old concept, even though some of us have forgotten it over the years. And I think building on that, and you guys will be really familiar with this, is that exercise is now quite commonly accepted to both prevent and treat a range of diseases, and lots of things that, not that long ago where, doctors would have been prescribing rest, even things like, people having cancer treatments or even something like pregnancy, where people were told to rest after heart transplants, things like that. People are now getting, prescribed exercise during chemotherapy, even in the days immediately after a heart transplant. So I think it's coming really well accepted that exercise is a really powerful medicine. If you were to ask people, I think most people would, agree with this statement that exercise is good for you. But despite that, everyone, it's quite obvious that there's not a lot of people aren't doing enough exercise, and these are just some numbers from Australia. But if you were to multiply these, probably put in a zero on the end, you're going to get something similar in America. So this was a survey, and I've adjusted the dollars, but in Australia, it's been estimated that insufficient fitness continues to cost the Australian government nearly $1 billion a year. And what's interesting, especially for people like us who are interested in exercise, is that exercise is quite cheap, but has really big benefits. So we had an economics company so Deloitte, you might be familiar with in America as well, I think, and they calculated that the cost effectiveness of prescribed exercise is about $600 per year per person. So that's having a trained clinical exercise physiologist who's prescribing the exercise for someone with type 2 diabetes with a well-being gain of $8,000 a year. So for quite a small investment, there's a big gain in health to be gained out of exercise. And I think that the medical community is starting to catch on, and sometimes see words like this bandied around. So this is the Academy of Medical Royal Colleges that's in the UK, United Kingdom, described physical activity as a wonder drug. And you see people like, I saw your alumni, Frank Booth, talk about exercise being a multi-pill, being able to target lots of different organs and systems and conditions. But one of the things that I'm going to talk about today, and what I think is a little bit sometimes missing, is a more precise exercise prescription. And if you think about, I don't know if it's a great example, but at the moment we're talking about the pandemic at the moment, but you wouldn't tell someone to go and get a moderate amount of vaccine. There's a very precise dose of vaccine at different times that people are given, whereas sometimes with exercise, I think even we're moving that way, more training type principles, but there's still a little bit of a gap there, I think, in having a more precise exercise prescription. And even when there is a prescription, quite often the target is so big that it's almost impossible not to hit the target. And so this was not a scientific article, this was in like a newspaper type article, but they were quoting this research and saying that moderate to vigorous physical activity is the most efficient way of boosting fitness. And to me, that's not a particularly precise prescription to be giving people. Oops. Sorry. Where are we? And I think just to highlight that a little bit more, what you can see here is, if we think about the moderate to high or vigorous exercise intensity, depending on the person, their age, their fitness levels, etc., the rating of perceived exertion could be anything from 4 to 18, and the percent of heart rate max, anything from 40 up to 100% of heart rate max. So again, even though we are moving towards, more detailed exercise prescription, I think that there's still, room to make those prescriptions even more precise so that we can get even better benefits for our health out of exercise. Okay. And so what I'm going to talk to you today is how we can maybe do better or move towards a more precise exercise prescription, so that a more individualized exercise prescription, so that more people can get the best benefits out of exercise. And the focus today will be on the mitochondria, obviously the energy powerhouses of our cells that supply the energy that our muscles need to contract, that we need to breathe, etc. But they also have lots of other roles in aging and cell function. And one of the reasons I guess we've focused on mitochondria in my laboratory, one being that obviously you need energy for everything, whether it's walking to being an elite athlete. But suboptimal mitochondrial function has been linked to a range of health problems. And some of them are pretty obvious. I know you guys do some work on diabetes, and so I think that makes sense. But some of the others you can see here, especially the ones in orange, are maybe a little bit less obvious. So even things like Parkinson's, Alzheimer's, Lou Gehrig's disease, or MND, have also been linked to suboptimal mitochondrial function. So I guess our thinking is that if we can use exercise to optimize mitochondrial function, then we can help to prevent and treat some of these diseases. Just to give you, a few examples, we can see here that I can't tell. The Zoom controls there? They're not covering any of the slides?
No, we can see your slides.
Okay, good. No, just on my screen, I've got the Zoom controls that are covering some of the things. I just wanted to make sure. So a bunch of studies have shown in different diseases, this one is elderly with insulin resistance, that there's when they compared with healthy controls, that there was about a 40% decrease in the rate at which mitochondria could synthesize ATP. And what's interesting is that with exercise, this is a study from a French group, but showing you can get similar improvements in mitochondrial function. So I guess providing some hope that with exercise we can reverse some of these defects. And we've also done using a different model, but we just published one of my previous PhD students with sleep. And so what we had here was a group, so NS was a normal sleep group, SR is sleep restriction, so this group had only 4 hours sleep a night for 5 days. And then we had another group that did exactly the same sleep restriction, so 4 hours a night, but they also did daily high intensity exercise. And what you can see here is that even just with only 5 nights of not getting sufficient sleep, we saw a significant decrease in sarcoplasmic fractional protein synthesis rates, which is an indicator of mitochondrial protein synthesis. And parallel with that, we also saw a significant decrease in mitochondrial respiration. So the ability of the mitochondria to use oxygen to produce ATP. And what was interesting is that we could prevent these defects by having daily exercise. So I get this research sort of highlighted two things. One, how the lack of sleep can decrease mitochondrial function, but also how exercise may be an option to prevent some of these defects. And parallel with that, these are healthy people, but again, after only 5 days of lack of sleep, we saw that these mitochondrial defects were also associated, so we did an oral glucose tolerance test with an increase in glucose area under the curve, so as an indicator of insulin resistance. So just a few examples of, and I think there's still a little bit of debate there about exactly whether to cause and effect, but there's good evidence that suboptimal mitochondrial function is related to a number of diseases and conditions. So there's been, for a long time, I guess, interest in the effects of exercise on mitochondria. And probably one of the main studies that is cited is this one of the first studies is this one by, I guess, quite a legendary professor in the field of exercise physiology, is John Holozzi. And what he showed in exercising rodents, that we could get quite a large increase in mitochondrial protein. So I guess that was as a marker of changes in mitochondrial content. What's interesting, I think, and I'll briefly mention today, is that this is held up as the first study, but we've actually tracked down almost 20 years earlier, or nearly 30 years earlier, is a study by Olga Cheponoga, who was a Ukrainian scientist. And in rabbit muscle, she showed exactly or similar thing, that after only mostly 2 weeks of exercise training, there was nearly a doubling of mitochondrial respiration in the rabbit muscle there. So we've known for quite a long time that exercise can, at least in rodents and animal models, that exercise can improve mitochondrial content and mitochondrial function. What our group does, and you may be familiar, so we use mostly humans in my lab, and so we use the muscle biopsy technique, where we take sometimes a reasonable amount of muscle, and we can then use that to do either ex vivo measurements of mitochondrial function, or we can look at mitochondrial proteins, mitochondrial genes, signaling pathways, etc. So most of the research I'll talk to today will be the human biopsy research that we do, and focusing on changes of mitochondria in the skeletal muscle. So where I'll start with this, and one of the I guess controversial and quite a debated topic is the role of exercise volume on changes in the mitochondria. And a couple of years ago, Professor Marty Gibala and his group and my group, we participated in a crosstalk in the Journal of Physiology. And so what we were, I guess, debating there is the role of exercise intensity and exercise volume in stimulating changes in mitochondrial content. So quite a important exercise prescription principle. If we're trying to increase mitochondrial content, how should we be going about that? And I think most people would be really familiar. Marty's done some awesome work on high intensity interval training, and so that was the position he took. This was the graph that we included in our position, and at the time we'd graphed, I think, all of the studies that had been published up until around about the crosstalk, and what we see is a decent correlation between exercise volume and changes in citrate synthase activity on the y-axis here as an indicator of changes in mitochondrial content. And as I said, you can see there a reasonable correlation, but there's also, if you look down at the individual studies, there's obviously a little bit of there's some differing results, as you often find in the literature. And you can also see we've highlighted there's not too many studies have actually compared different types of exercise within the one study, but with the colors there, we've highlighted a few of them, and you can see examples of both. So you can see here in one of Marty's studies that the high intensity exercise had a much greater improvement than the moderate intensity continuous training, whereas we've shown the opposite, and here again the opposite there. But when we looked at it I think, as a trend of trying to look at all the studies and I guess trying to minimize some of the variability between the different approaches of the labs, our position was that we saw the greater the exercise training volume, the trend is for a greater improvement in citrate synthase activity. It just reminded me that this might be a good, I think most people are familiar with the terms, but these will be popping up quite a bit through the talk, so I should just briefly mention them. So if you see MICT, what we're talking about there is moderate intensity continuous training. So that's typically continuous exercise, going for a 5-10k jog, for example, would be a good example. Typically between about 50 and 80% of VO2 max, certainly below the lactate threshold. We've also got HIIT, and this sometimes different labs use different definitions, but what I'm referring to here is high intensity interval training. And so we're looking at intervals typically performed between about the lactate threshold and VO2 max intensity. And then we've got SIT or sprint interval training, which is above, at least as we're using it here. So briefer intervals, so maybe around about 20 to 30 to 40 seconds at an intensity that's above VO2 max intensity. Typically that's going to be an all out, but it's not always, as long as it's above the VO2 max intensity. So apologies for that quick detour. So just saying, so with our position was that greater exercise volume leads to greater improvements in this marker of mitochondrial content, citrate synthase activity. It ended up being one of, I guess, the most commented on crosstalks in the Journal of Physiology. And if you have a flick through it, there's a bunch of esteemed scientists from around the world. Some people agreeing with Marty's proposal that exercise intensity is more important to increase mitochondrial content, and probably about half agreeing with us. So it's still probably an unsettled topic in the literature. One of the things that I think it's important to point out, and I can talk about this in more detail later if anyone has any questions, but it's important to point out that the measure of mitochondrial content that's being used here is citrate synthase activity. And so what we're talking about is the activity of one protein in the mitochondria, which consists of, and I think we don't really know exactly, but somewhere between 1,200 and 1,400 proteins. So I think that there are some legitimate questions about whether changes in citrate synthase activity accurately reflect changes in mitochondrial content. And I suspect sometimes with the high intensity, especially with the very high intensity training, it may be that there's an increase in the activity of the enzyme, which doesn't actually reflect changes in the mitochondrial content. And we've just published a paper in Nature Communications, which I think illustrates this really nicely. There's lots of data in that paper, but one of the main things which was surprising that we found is that the different mitochondrial proteins changed in different ways and not always in the same direction. So, and we found that there was basically 6 different clusters. And so what we've got here, this is one group of mitochondrial proteins, which I think was beta oxidation, which increased early on, as did the TCA proteins. But we actually saw, relative to the changes in mitochondrial content, a decrease in OXPHOS proteins, complex Q proteins, and also proteins of cristae remodeling as well. So sometimes I think there's this idea that all of the mitochondrial proteins go up and go in the same direction. But what we've seen is that depending on, we've had 3 different phases of training, but different phases of training, you can have very specific mitochondrial adaptation. So that's kind of just a caution against being, I think just being cautious with our interpretation of changes in citrate synthase activity and whether they reflect actual changes in mitochondrial content. Nonetheless, I think, regardless of what we use as our measure of mitochondrial content, so we've used micrograms of protein in our mitochondrial isolates, we've used citrate synthase activity, and we've also used what is here is the summed intensities of our mitochondrial proteomics data. We always see the data going in the same direction, and that is when we use the highest volumes of training is where we get the greatest changes in any marker of mitochondrial content. So again, we still maintain that exercise volume is important to get the greatest changes in mitochondrial content. And you see this also if we use electron microscopy, transmission electron microscopy. So that's where you're doing actual images of the mitochondria. And this is where we've summarized the data from the 6 or 7 studies that have been published. And what you see with this measure of mitochondrial volume density, so this is an imaging method, doesn't rely on an enzyme. The larger the training volume, the greater the increases in mitochondrial volume density. Although there's only 2 studies here, but it appears that maybe there is a plateau, and that actually, I think that makes sense from a training perspective as well, as that you wouldn't expect that a linear increase would continue forever. But if you look through this part, so even when we use imaging, that seems to support as well that the greater the training volume, the greater the improvements in mitochondrial content. And there's been very few studies that have directly compared different types of training on changes in mitochondrial content using TEM, or transmission electron microscopy. And so this is a study from a previous PhD student that we're completing the manuscript at the moment. And what we did is we directly compared sprint interval training with moderate intensity continuous exercise. So the sprint interval training was about 30 minutes, but with 6 30-second all-out sprints, whereas the continuous exercise was below the first lactate threshold for 90 minutes. And so we purposely did it so that the orange is the sprint interval training and the black is the moderate intensity continuous exercise. The volume was about 5 times greater in the moderate intensity exercise, and the intensity was about 5 times greater in the sprint interval exercise. And you look over here, using the TEM to directly measure mitochondrial volume density, we didn't see a significant increase in mitochondrial volume with the moderate intensity continuous exercise, and we only saw a significant increase when we did the sprint interval training. And that was actually also reflected in our citrate synthase activity as well. So one of the questions that then arises is, from a mechanistic point of view, what might it be about training volume that is important to increase mitochondrial content? And we really don't know, but one of the things we've been looking at is the role of muscle glycogen. And this is from a review that we just published late last year. And what we've seen is that and this is again, we did like a meta-analysis of the published studies, but there seems to be a relationship between glycogen reduction and activity of AMPK. So maybe with the greater volume of exercise, we can get greater reductions in muscle glycogen, which then start some of the signaling cascades that result in mitochondrial biogenesis. And a lot of you will be familiar with this, with I guess the train low literature as well. And so this is from Kent Sarland's group in Sweden. And what they showed is when you did continuous exercise, but with a lower muscle glycogen, then they saw a much greater activator of PGC1 alpha, which commonly termed the master regulator of mitochondrial biogenesis. And Louise Burke and John Hawley published a really nice review in Science on this a few years ago. And so it appears that training with low glycogen, and that can be by depleting muscle glycogen levels overnight, or we've also seen by doing glycogen depletion in the morning followed by an afternoon session a few hours later. By decreasing the muscle glycogen, it appears that we can increase the activation of these pathways that lead to muscle glycogen, to mitochondrial biogenesis. I'm sure there's other mechanisms, but this is one potential explanation for what we see as the powerful effect of a high training volume. So just in summary on that section, so there's definitely a lot of debate around, but what our research and that of others appears to be showing is that if you can to get the greatest changes in mitochondrial content, then it's important to do a high training volume. I can just see someone's put something in the chat, so I'll just check there. Sorry, Alan, I missed that before. If it's okay, I'll come back to. I can mention that quickly. So when we did those sleep studies, we were doing high intensity interval training, and it was based on a protocol by John Little had used in Canada. But it was high intensity interval training that I think lasted around about 20 to 30 minutes in total. Okay, let me get rid of that. So I've just been discussing this. So I think, as I said, I think there's pretty good evidence, at least in our hands, that greater training volumes seem to be a powerful determinant of changes in citrate synthase activity. What's interesting is when we take the same data and look at the relative intensity, we don't see any, you can see there's a flat line there. So we don't see any relationship at all. And so we would argue that training volume is more important than training intensity. Sorry, I just saw it. Thanks. No worries, Alan. But then when we look at changes in mitochondrial respiration, in this case, mass-specific mitochondrial respiration, it appears that there is from the few, there's not that many studies there, but there appears to be a window that to get the greatest changes in mitochondrial respiration requires high intensity training between about 80 to 100% of VO2 max. And I should just pause here and explain a couple of things, or define a couple of things as well. So you may see this term pop up in the literature and in our studies. So we're looking at, when I talk about mass-specific mitochondrial respiration, that's when we express the respiration relative to the amount of muscle mass that we're using in our analysis. So that it's probably in some ways analogous to an absolute VO2 max. So people with a greater muscle mass will have a greater respiration. But so this is a common term that's used there. And that perfect question, and I'll pause there as well. So Fataha, apologies, hope I pronounced that okay, has asked, does higher mitochondrial content always translate to higher mitochondrial function? And the simple answer is no. And so that's why I'm dealing with these slightly differently, because we've seen examples where we can get quite different responses. So depending on the exercise, you can get improvements in mitochondrial function without improvements in mitochondrial content, and also vice versa. Sometimes they will occur in parallel, but they don't necessarily. So just sorry to jump back to here. So what we've seen here is that it appears that the intensity of training is really important to improve, in this case, mass-specific mitochondrial respiration, which is a measure of the ability of the muscle mass to use oxygen to produce ATP. What we do see, though, is there's another way of looking at this. And so what I've referred to here is mitochondrial specific respiration. And so what we do there is we take the respiration measure and we normalize that to the amount of mitochondria that we have in our sample. So this isn't exactly what it is, but just as an analogy, you can think about it as the ability of each mitochondria to be able to use oxygen to produce energy. And what we see when we look at this measure is that the sprint interval training is a really powerful way, and much more powerful, to increase the mitochondrial specific respiration. And this probably also answers Fatiha's question, because what you see here with the moderate intensity exercise training and the high intensity interval training is that the increases in mitochondrial respiration parallel or in proportional to changes in mitochondrial content. So the changes in respiration are mostly due to an increase in mitochondrial content. However, something different is happening when we're doing sprint interval training, and we don't see large changes in mitochondrial content, but we do see much larger changes in mitochondrial specific respiration. So the ability of each mitochondria to produce ATP. As before, the question then becomes, why is it that specifically the very high intensities seem to be important to stimulate the adaptations and improvements in mitochondrial function? And the last few years, and I'll present some unpublished research where we've looked at some of these, some of the potential mechanisms or reasons to help explain the powerful effect of high intensity exercise. And one of the theories is that because of the Henman size principle, is that when you're doing high intensity exercise, you're going to be getting greater recruitment from the 2A and possibly also the 2X muscle fibers, and maybe that's contributing to some of the greater changes in mitochondrial function with the sprint interval training. So we've just almost, hopefully it's really close to submitting this paper that we completed, this project we completed last year. But I'll run you through the experimental protocol. But basically what we had is 24 young men. They had a resting biopsy. They came and then did two types of training that I described before. So they did either 8 weeks of moderate intensity continuous training, which was 4 days a week of 90 to 120 minutes of continuous exercise, or they did 8 weeks of sprint interval training. We then took a post-training biopsy. We then isolated the single fibers from the muscle biopsy. We then digested those fibers, and we did a process called dot blotting, which we used the myosin heavy chain antibodies to identify whether each individual fiber was either a type 1 or 2A muscle fiber. Based on this, we then pooled the single fibers so that we then had 6 type 1 fibers or 6 type 2A fibers. We then processed those and we labeled them, and then we did mass spectroscopy, which is a way for us to look at, I think on average we're able to detect 2,000 proteins within each single fiber, and round about that was 400 mitochondrial proteins as well. And then when we looked at the results, so you can see here, and this is just a summary of the proteins that we're able to detect that were either complex 1 through to complex 5 and also the mitochondrial ribosome. But if you look on the right here, you see exactly, I guess, what we would have hypothesized. So the blue is the type 1 fibers and the red is the type 2 fibers. And we only saw increases in the mitochondrial complex proteins in type 1 fibers. And yeah, there's a little bit of variability, but there was very little change in the mitochondrial proteins in the type 2 fibers, which, is what we would have hypothesized, because of there shouldn't have been much recruitment of the type 2A fibers. So that was nice to see. What was surprising, so when we look at the left here, so this is with the sprint interval training, we actually we saw, if anything, a decrease in the mitochondrial proteins. And we suspect that this is probably coming back to the training volume that we were talking about before. And even though it was very intense, it was only 30 seconds times 6, so 3 minutes of very intense exercise, is that the training volume was not sufficient to get any large changes in either the type 1 or the type 2 fibers with the sprint interval training. And we also did what these profile plots, and I think they highlight it quite nicely as well, some of the other processes. So I've got moderate intensity training on the left and sprint interval training on the right. And you'll see for each color, there's 2 rows. So the first row is the changes in the type 1 fibers, and the second row is the changes in the type 2 fibers. So you can see the same pattern where proteins associated with fatty acid oxidation only increased in the type 1 fibers with moderate intensity training, and there wasn't really much happening in the sprint interval training. And that pretty much gets replicated for proteins involved in mitochondrial dynamics, so that's fission and fusion and cristae remodeling, and same with the TCA cycle. And interestingly, when you look at glycolysis, that's where we see the opposite. And so you're seeing a decrease in the glycolytic proteins, but only in the type 1 fibers with moderate intensity training, and not too much happening in the other situations. We're also interested in this idea that, one of the reasons for some of the adaptations we see with the high intensity exercise is because of the greater stress on the muscle. So the homeostatic perturbations that occur when you do high intensity exercise, and in particular the changes in lactate that occur when you do high intensity exercise. So what we did in this study, which is still unpublished, is we had 10 guys come in. They were quite well trained, and they came in on 3 different occasions, and in a random order they did either low intensity exercise below the lactate threshold, or they did moderate intensity exercise between the lactate threshold and the maximal lactate steady state, or they did high intensity exercise which is above the maximal lactate steady state. And we took biopsies before and after exercise. And just to show here that we achieved what we wanted in terms of quite distinct blood lactate profiles. So the low intensity, there was very little change in blood lactate. The moderate intensity, there was a slight increase, but you then reached a steady state. And when we went above the maximal lactate steady state, there was a continual increase in the blood lactate level. So we tried to get 3 quite different homeostatic perturbations. And with the muscle biopsy, again, we isolated the RNA, and we then did RNA sequencing to be able to look at tens of thousands of genes that are potentially altered with exercise. And as I said, we're in the early days of analysing this, but the interesting thing, these numbers here are the number of genes that were changed after exercise. And when you do low intensity exercise, if you're able to add up all those numbers in your head, but you don't need to. Either low intensity or moderate intensity exercise, we see a similar number of genes that are altered. But once we go above the maximal lactate steady state, we see almost a doubling of the genes that are activated or that are altered with exercise. So we're now exploring exactly which genes, but I think this just highlights that the potentially there's something that the exponential increase in the homeostatic perturbations that occur above the lactate threshold lead to a disproportionate change in the number of genes that are altered with exercise. And there's a lot of research pursuing, I guess, there's other things than lactate that obviously change above the maximal lactate steady state, but some of the pioneering work of George Brooks and other researchers since, I think, has highlighted that the lactate may be an important signal that helps to translate the different exercise intensities into adaptations. And so, Hashimoto in George Brooks's lab kind of kicked off this area of research, showing that lactate, so they added lactate to cell culture, produces ROS, which seems to increase the binding of ROS responsive transcription factors, which will then increase transcription of genes such as PGC1 alpha. A really cool paper in Nature a couple of years ago showing that the addition of lactylation is what they talk about. It was an important histone modification caused by lactate, which also increases transcription. And we've also shown that if you add, this is again in cell culture in L6 cells, but if you add lactate to cells, you get a decrease in nuclear HDAC. So this is an important protein that maintains chromatin in a condensed format, which makes it hard for transcription factors to bind. But if you can remove HDAC from the nucleus, then that leads to a more open chromatin, which then increases transcription as well. So again, this is a topic that our lab's quite interested in, is the role of different exercise prescriptions and the response of lactate and how that may translate different exercise prescriptions into different adaptations. And one of the other, not only, I guess, there's a homeostatic stress in terms of changes in lactate, ADP, glycogen and things like that, but there's also evidence that exercise also has a direct stress on the mitochondria. And so these are some TEM images that we took after sprint interval training. And I should have put up, or you would have saw, from one of the earlier pictures, but I guess a normal healthy mitochondria. But you can see here, we've got some images. This is in muscle biopsies taken after sprint interval training, that we've got some quite damaged mitochondria here. You can see the cristae isn't the beautiful sort of classic mitochondria, and here you can't see really any cristae as well. So this is these are quite damaged mitochondria, and basically we only see them after sprint interval exercise. So this is all the cristae were normal when we took biopsies after moderate intensity continuous exercise, but we saw nearly 15, nearly 16% of the mitochondria showed some form of damage after sprint interval exercise. Maybe, and kind of seems maybe it's not ideal, but if you think about it also, with resistance training, you get some of the micro tears in muscle which are responsible for help to start the process of repair and building new muscle. So it may be that this is a good thing, and maybe this damaged mitochondria is providing an important signal that helps to explain why sprint interval training can increase mitochondrial function without changes in mitochondrial content. And in these same studies, we did some RNA-seq again, and you can see very different activation patterns between the moderate intensity. So if you just look at the red and the blue as indicator of genes that are either down or up regulated, so down in the red and up in the blue, even if I didn't have these labels up here, you could see that quite easily that there's something very different going on with these different types of exercise. And so I guess coming to back to the point of exercise prescription, it seems to be, it's really clear that different exercises are activating different transcriptional pathways and also leading to different adaptations. One of the ones that was quite interesting to us is in this bottom right-hand corner here. And so sprint interval training very strongly activates what's called the mitochondrial unfolded protein response, which is going to be important to lead to the mitochondrial biogenesis. And we've also verified this with PCR. And so these are some targets of the mitochondrial unfolded protein response. And you can see here in the orange that the sprint interval training in all cases is leading to a greater activation of these genes than the grey, which is the moderate intensity exercise. And running out I'll finish up here, but I think the other interesting question at the moment is, we talked about high volume training and high intensity being important for different aspects, but is there a point where you can do too much exercise? And this was been quite a big debate at the moment. So this is a paper that got a lot of interest last year from Sweden, from Flockhart. And what they argued was that, so in orange, when they did what they called excessive training, which was 5 high intensity interval training sessions a week, they saw a decrease in mitochondrial respiration measured in isolated mitochondria. So really interesting finding, but we don't see that, and even with more severe exercise. So I've tried to graph it similarly to what they did. So we did 3 high intensity interval training sessions a week for a similar number of minutes as what Flockhart did, but we then did massively, nearly 4 times as much high intensity interval training as they did. So we did high intensity interval training twice a day, 7 days a week for 3 weeks. So you can see nearly 600 minutes of HIIT in a week. We saw significant increases in mass specific mitochondrial respiration, but we didn't see any changes in mitochondrial specific respiration. But we definitely didn't see a decrease as they argued. And I think one of the important, I'm not doubting their results, but one of the important things to note, so they've done isolated mitochondria, whereas we've done mitochondria in permeabilized fibers. And with John Hawley, we wrote a viewpoint in Nature Reviews Endo at the end of last year. So it's important to remember that, when you're measuring VO2 max or something similar, what you're doing is you're measuring in part the ability of the mitochondria inside the muscle to take the oxygen from that delivered from the lungs in the circulation and to be able to produce energy. When we do respiration in permeabilized muscle fibers, we're taking obviously the muscle fibers out of the body, but we're still keeping the mitochondria in the fiber, and we're just permeabilizing the sarcolemma so that we can get substrates to the mitochondria. The other measurement that was used in Flockhart is to actually isolate the mitochondria and take them out of their muscle fiber, so they're just sitting in solution. And we know that you get less than 20% of the mitochondria when you do that, and we know that a lot of them also get damaged as well. So it's just a cautionary tale about, depending on the model that people are using, are isolated mitochondria actually representative of how the mitochondria are functioning in an integrative physiology sense in an intact human? So I'll let you just quickly summarize the what we've said there. So both high intensity and high volume exercise training are effective at increasing mitochondrial content. However, evidence suggests that training volume is more important to produce the largest changes in mitochondrial content. High intensity, high volume training appears most effective to increase mass-specific mitochondrial respiration. Very high intensity training, such as sprint interval training, appears most effective to increase mitochondria-specific mitochondrial respiration. And more research is required to understand the underlying mechanisms for these divergent adaptations. Just quickly, obviously, I've got an amazing team, some awesome PhD students and postdocs who do a lot of this research. So thanks to them and the funding agencies. And thank you. And hopefully that's given you a little bit of an idea how we can perhaps better prescribe exercise to improve health. Thank you.
Thank you so much. That was a really, really interesting talk. And I finally saw someone, I mean, I've never actually seen a lot of people use Prezi, so it was nice to see you use it because a lot of us still stick with PowerPoints and so that was really cool. Thank you.
Yeah, pleasure. I see. Thanks for Megan's comment on the accent. Can't do. And from Nadine, how would you translate the significance of your findings in general sense clinically for exercise prescription and improving overall health? Yeah, I think it's interesting, these questions, isn't it? It's tough. I think we're still on I hate to say this, the more research thing, but yeah, I think we are seeing that different prescriptions do have different adaptations. So I think we're still trying to work that out there. But I think, I think a good example is Marta Murgia gave a talk to us on Wednesday. Is even the fiber specific stuff. So what you see with aging is obviously you get general atrophy, but in terms of some of the mitochondrial proteins, you especially see decreases in the type 2 fibers. And so I think, one of the things that's come out of her work and what we're interested in is, with the elderly, quite often the prescription might be general physical activity, and sometimes it might even be gardening or, quite low intensity activities. But I think, those activities clearly aren't going to activate type 2 fibers. And so, I think that's kind of made us realize that, we do really need to think about, if we want to maintain the mitochondrial health in all those fibers, we do need to think about prescribing higher intensity exercises to those who are capable. We also see, there's with diseases, different aspects, so diabetes, different aspects of mitochondrial function that are decreased with those diseases. So I think that's what I mentioned before. I think there's going to be some, and we're not there yet, some diseases where it's probably going to be more important to increase mitochondrial content. So that might require a more specific exercise prescription. Whereas there's going to be other conditions and diseases where maybe increasing mitochondrial function is more important. And in those cases, it may be more important to prescribe higher intensity exercise. But I think even on maybe even a more basic level is, I like to think about, especially in terms of the mitochondria, the dietary pyramid is, I think most people are really well familiar with that. But I think exercise, we need to be thinking about that a little bit as well. So we're doing at the moment some studies where, the classic experimental protocol is to do either one or the other. But given our results, we're looking at what happens when we start to integrate these results. So can we do a more complete program that involves continuous exercise to target mitochondrial content, but also introduce some sprint interval training to target more changes in mitochondrial respiration. So, I think it's always incremental, but I think what we're trying to move towards is, a more accurate prescription to be able to get the mitochondrial adaptations that are needed for different individuals and different conditions.
Yeah, thank you.
Okay, someone DKB just asked that. I just answered that. Yeah. So yeah, I agree. I mean, I think if you, you know, like I said, in nutrition, you wouldn't sort of recommend just protein or just carbs or whatever it might be. So I think the training is the same. That hopefully we're moving towards it where we can give a more precise, you know, prescription about how to best incorporate those different elements. Fatih has asked about the best measure of mitochondrial function. I think at the moment, I mean, with some bias because it's what we do in our lab, where I mean we're measuring we definitely do permeabilized fibers. So I think that's best in terms of the mitochondria is inside the muscle fiber. It's attached to the actin and myosin filaments. It's got the, you know, everything else that, you know, the other substrates, etc., so that it can function more normally. But I think what we're moving towards, and there's some really nice work from Graham Holloway that we're collaborating with in Canada, is I think, you know, sometimes if you took so we quite often measure maximal mitochondria, so we're measuring the maximum capacity of the mitochondria to use oxygen to produce ATP, but I've seen some estimates, if you took the mitochondrial function measurements and extrapolated them to a whole body, you'd get something like a VO2 max of 200 or 300 mils per kg per minute, which obviously doesn't, nobody has that VO2 max. So I think the mitochondrial capacity is way in excess of what it needs to be. So I think there's some question marks about whether the mitochondrial function, the maximal respiration is the best measure. So we're looking at the ADP kinetics now increasingly, which is how quickly does mitochondrial function increase in response to a decrease in ADP. And I think that's probably going to be a better measure of mitochondrial function. I can see Thaisa, nice to see you, bon dia. Good question. We did some work with Thaisa's got some really nice work on carbohydrate and signaling and mitochondrial function that we did in Brazil. We haven't looked at it in terms, she asked in terms of patient groups, but I think it's a really good question, and I think that's what we're increasingly going to see. I saw Julien Zirath, who's a great, unbelievable scientist from the US, but in Sweden now, and she just wrote an opinion paper on, I can't remember the exact title, something like Train Like an Athlete. But I think we will start to see more of these. I think athlete training models filtering down to normal or disease populations. I think the only caveat I would say there is I think a lot of what athletes do is kind of like the icing on the cake, whereas with with people with cardiovascular disease or diabetes, their aerobic fitness is so low and their mitochondrial function is so low. Getting them to exercise and getting them to exercise well is probably going to be the priority. And I think adding, whether it's hypoxia or glycogen depletion or some sort of supplement on top of their exercise training, I think that's probably that's kind of the icing on the cake that athletes need when they're plateauing and getting close to their limits. But I think, I may be proved wrong, and maybe it's going to be an efficient way to quickly improve some of these populations. But most things that I see is exercise is really the most powerful stimulus. And whenever you manipulate nutrition and things like that, it's able to tweak and add a little bit on top of the exercise. But the exercise, to use the analogy of the big rocks and the little rocks in the container, the exercise and the exercise prescription, I think, is is the big rock that's going to have the biggest effect.
Any more questions for Dr. Bishop? All right, well, thank you very much. We've taken up a lot of your time, but this was such an excellent talk, and thank you for accepting our invitation again. I'm already hearing from people, everybody loved it. Thank you. And if everybody could just unmute your mics, let's just give Dr. Bishop a round of applause.
Thanks a million.
It was a pleasure, and appreciate the invitation. Have a
Okay, thank
good evening.
you.
See ya.
Bye.
Bye.